project we had with Innovate is ongoing at the moment, and that's looking at oligonucleotides, synthesis and purification. Our technical challenge was to develop an agarose-based chromatography media which could be used at higher temperatures and to create a column system which would maintain those temperatures accurately. The funded project was to look at mRNA production, manufacturing through in vitro translation. So we were looking at how we could reduce the cost of goods by reusing some of the most expensive reagents. We make agarose beads which have a shell on the outside and an agarose core. The chemistry that we've used allows you to then change the conditions of the buffer and actually wash out those reagents and capture them, ready to reuse for the next step. The other part of the project was to look at separating single and double-stranded RNA without having to use high-pressure chromatography systems or solvents. So again, making it much more accessible, scalable, and much more environmentally friendly.